it's a great pleasure to introduce Sandra Diaz, who's a professor at the of ecology at Cordoba National University in Argentina, and she's also a visiting professor here in the in geography and with the Leverhulme Center for Nature Recovery. And she's just coming, and she's got a couple more weeks left this time, but she'll be back again the next 2 years for about 4 months every year as well. So it's really, really valuable to have you as part of our wider team for this. And she's done a lot of work, particularly in plant functional traits, and then created and coordinated the TRI database, and we'll hear more about that. But beyond that, she also has a major role in linking science to policy. She co-chaired the intergovernmental platform for biodiversity and ecosystem services, and provided major expert scientific advice on the Kunming-Montreal Protocol Global Biodiversity Framework. And she's a foreign member of the Royal Society, member of the American Philosophical Society, and the member of the Academies of Sciences of Argentina, USA, France, Norway, Latin America, and the developing world, and the American Academy of Arts and Sciences. So there's quite a collection. There's more there that I could mention as well, but that's good enough for now. So thank you for presenting to us, and over to you.
Okay, thank you. Good afternoon, everyone, and thank you for coming. This talk is intended as a stock taking, very personal stock taking of a field of knowledge that it has been in my focus of interest for a very long time. Plant functional traits and functional traits in general. Okay. So since the very origins of empirical ecology, there's always been this tension between, on the one hand, celebrating the myriad ways in which every organism is slightly different from the rest, and on the other hand, searching for a few general rules or patterns underpinning all this fantastic complexity. In other words, searching for the ways in which organisms are similar to each other. And since the 1990s, this long-standing tension, together with an increasing sense of urgency posed by the challenges of environmental change, fueled an explosive resurgence of what we call the functional trait research program. And this expansion is continuing to this day. The functional trait research program can be brutally summarized by this diagram. Functional traits, here represented by these colorful bits, determine an organism's responses to the environment and therefore its fitness. And functional traits also determine the organism's effects on ecosystem properties and the benefits and detriments people get from them. And one of the contributions of the functional trait research program to expanding the frontiers of fundamental ecology in general has been to show that there might be myriad ways in which every organism is slightly different from the rest, but there should be a very limited number of essential designs, a very limited number of combination of values of essential traits. Put it in another way, who of you has spent more than 2 days in Mexico, apart from Jesus, of course? Okay, many of you. So you probably came across the alebrijes. The story goes that there was this humble folk artist, Pedro Linares, and he carved wooden little animals for a living. And one day he fell ill, and because he was too poor to pay for a good hospital, he became worse and worse, and he became delirious and had this feverish dream in which he was in a forest surrounded by creatures displaying the most bizarre combinations of body parts of different animals. They had the horns of a bull with the wings of a butterfly, the beak of stork with the claws of a jaguar, etc. And they were all loudly screaming at him, alebrije, a word that until then didn't exist. And then Pedro woke up and started making alebrijes and became world famous because people love alebrijes with their brilliant colors and unusual combinations of body parts. But what I try to persuade you today is that nature doesn't like alebrijes. So when all these functional traits get packaged together into whole body phenotypes, they don't do so in orthogonal or random combinations. There should be a limited number of successful combination of these fundamental traits. Now, these ideas have been put in the literature on theoretical grounds a long time ago, and they have been tested for individual organs, but nothing further than that. So a few years ago, we pioneered the testing of these ideas at the level of the whole plant and at the global scale. What we wanted to get at is the three major aspects of the struggle for existence of any plant: growth, survival, and reproduction. And we wanted to identify the very minimum set of traits that would be fundamental enough to capture these three dimensions, but at the same time would be available or easy to measure afresh for a very large number of plants, of species worldwide. So we finally settled for these 6 traits. As you see, they are fundamental, but they are almost disappointingly simple. But simple as they are, it wasn't trivial to put together a data set of enough size. It took us years to do it. And we only were able to do it thanks to TRI. TRI, as some of you may know, is the world's first and so far biggest communal repository of plant functional trait data. And since I'm talking about TRI, I would like to devote a few moments to honor the memory of this guy, Gerard Bernish. Even if you are familiar with TRI, you are unlikely to have met him because he was a man of few words and he didn't go to many conferences. But he was the IT person at the pumping heart of the whole of TRI. So until he died a few weeks ago. So he's very badly missed by the whole community. So, as I said, we put together this very big data set. Even to this day, this is the largest functional trait data set for empirical data and data at the level of species. Despite the fact that it's so big, it only covers just above 10% of the total number of vascular plants species known to science. But we did manage to cover many families, many growth forms, and all the inhabited continents. And we also managed to get data, probably some of the most extreme values of these fundamental traits known to science. So we felt that we have covered the limits, the boundaries of trait space of vascular plants pretty well. With this data set, what we wanted to do is to build the observed global multifunctional space occupied by living vascular plants, what we call the observed global plant phenospace. We built the observed phenospace here, and we tested against a number of null models. We rejected all null models, meaning that the observed global plant phenospace is subject to strong constraints and trade-offs, and is very granular, very lumpy. In other words, there are not very many alebrijes in the plant world. When we explored this plant phenospace further, we found that as much as 3/4 of the total functional variation was captured by just one plane. This plane, which we call the global spectrum of plant form and function. This was the first global picture of essential functional diversity of vascular plants. It's, of course, a multidimensional space, but is rather flat, pretty much like a chapati bread. Now, I don't want to dwell too much here because the paper has been out for a few years now, and at least some of you may be very familiar with it. But I do need the whole of you to be on the same page because for the rest of the talk. So very briefly, this plane, the global spectrum, has two major axes of variation. One of them is this one. It's an axis of size. And it goes from very small plants with tiny leaves and seeds, all the way to tall plants with large leaves and seeds, from duckweed to Brazil nut. And the second major axis of variation, this one, is an axis of pace of life and leaf resource economics. And it goes from highly conservative plants that allocate resources preferentially to defense and support and have relative leaves that grow slowly and live long. Examples, holly, junipers, or even more conservative, the Australian hackers. And at the other end of this axis, there are highly acquisitive plants with a very strong allocation to growth, with leaves that are nutrient-rich, flimsy. They are displayed very quickly and they die very quickly, like basil, for example. Now, a couple of words about what this color map represents, because it's going to be the same for all the other graphs I show. This is a probability density map. And it shows how many times a particular combination of plant traits is found among existing species. So you can see here, there are some plant designs, some combinations of traits, which are much more popular than others. And this gives rise to a very lumpy phenospace with a very steep topography, with red hot peaks here that have a very, very large functional redundancy, lots of plants with more or less the same trait combinations, surrounded by much colder valleys here, populated by species which show much more rare combination of trait values. And these hotspots are not just the result of some splurge of speciation at some particular point of evolution. They are successful solutions found again and again over the whole evolutionary history of angiosperms. So, so far, I show you that the global phenospace of vascular plants is very constrained, oligodimensional, and lumpy. But this doesn't seem to be only the case of plants. In the years that follow the publication of our paper, a number of colleagues embarked themselves in similar search for distilled, simplified phenospaces for other organisms. So, for example, there is the pioneering work by Alice Bigot and Joseph Tobias and Helen Schert from the biology department with birds. And they show that the global phenospace of birds can be captured by 4 dimensions, and more than 80% of the variation is captured by the first 2 axes. And body size is very important in determining this global phenospace. And in the following years, there were many other spectra for many other groups being described in the literature, for ants, for corals, fungi, soil bacteria, bryophytes, and all sorts of animal groups, with different approaches, some of them more parsimonious than others, but this is a very active area of research. And there is, of course, some synthesis work. So this is coming, for example, from the great synthesis work on the basis of already published data sets by Carlos Carmona and Rob Salguero here, and other colleagues. And remarkably, they showed that groups other than plants also have phenospaces which are oligodimensional. In most cases, more than 70% of the total variation is captured in the first 2 dimensions. In most cases, these first 2 dimensions are strongly determined by body size and pace of life. And virtually in all cases, the phenospaces are definitely lumpy, with some combinations much more popular than others, marked here in the hotspots. And more recent work by more or less the same constellation of people sort of reinforced these findings. So this is the work by Carlos Carmona and a bunch of colleagues, including myself. What we did here was to add fine root traits to the global spectrum of plant form and function. And fair enough, we found that root traits add 2 non-redundant dimensions to the plant phenospace, but these dimensions are definitely subordinate with respect to the first 2, which are still prominently body size and pace of life of the other above ground plant. And very, very recently, this time for animals, there is the work by Eleonora Beccari and the Carmona group and the Salguero group with mammals. And they found that the global mammal phenospace can be captured by 2 dimensions, dominated by the slow-fast continuum here. And the second dimension has to do with heteroparity versus semel parity, and the degree of maturity of the offspring at birth. You see here, there's no body size in this phenospace, but that's because the authors, after very explicitly acknowledging that body size is overwhelmingly important, chose to standardize all traits by body size prior to analysis. That's why you don't see it here. I think that taken together, all these findings point to the existence across kingdoms of a fairly limited number of predictable trait combinations that include but go well beyond those expected solely on the basis of shared ancestry. And body size and pace of life is almost universally important. Now, I take two very general reflections from all this. One is that nature doesn't only dislike plant alebrijes, it seems to hate alebrijes in general. And the other one is that the magnificent Baroque variety of life on Earth seems nevertheless amenable to very strong distillation by means of quite simple essential phenospaces. Now, by now, you may be asking yourself the next obvious question. That's fine, but aren't these distilled phenospaces far too simple? And the short answer is probably yes, they are. And that's why they can be really useful. These phenospaces are too simple for a number of reasons. One of them is that they typically build with the global mean of the trait value for each species. And we know that trait values vary a lot between individuals of the same species across environmental gradients, among individuals of the same species on a site, even across the life stages of the same individual. But doesn't the variability at one level doesn't necessarily invalidate variability at other levels? The important thing is that the differences between the 2 points you choose to compare are still meaningful. So for example, here, the numbers represent 3 species projected on the global spectrum and on the basis of the global mean trait values. Olives, species of wheat, and Arabidopsis. And the smaller circles represent the positions of individual populations or accessions of the same 3 species. And you can see in all 3 cases, there's quite a scatter, quite a cloud around the global mean. This may be a bit extreme because in all cases, these populations have been deliberately bred to be as different as possible. But in any case, it's undeniable that in nature, there's a lot of intraspecific variability in traits. But I would argue that the global spectrum, or when we talk about animals would be the same, doesn't deny this intraspecific variability. It just simply states that the global means are still informative. So in a way, it's like zooming in and zooming out in a map. For example, if you are trying to find your new flat in an intricate neighborhood, you will be probably using, I don't know, Google Street View or something like that. But you wouldn't be choosing to use Google Street View if you're trying to start planning your overland trip across Africa. It just wouldn't be practical. Another way in which, well, another reason by which these highly distilled phenospaces can be accused of being too simple is because they typically involve very few dimensions. So the question here is whether a brutal splatter onto 2 dimensions makes justice to the variety of life on Earth? And the short answer is again, no. That's why probably a number of colleagues have busied themselves in the last few years trying to unpack spectra for organs. They are trying to produce spectra for seeds and for flowers and fruits and roots and stems. And I'm sure that while we are here, somebody is trying to come up with the global spectrum of leaf trichomes or the global spectrum of ear shapes of rodents, right? And this is all fine. This is all fine because it adds to the richness of our understanding of life on Earth. And of course, the more traits you include, the more precise your picture is. But the key question is whether this extra effort is worth the precision we win, according to your question. Because including extra traits is never, ever free. The more traits you include, the more theory you need to have to support your choice, the more you struggle trying to run your models, and the harder it gets to get a full trait empirical coverage for your species. So you are faced with a challenge between 2 temptations. On the one hand, reduce the number of species, but then you still want to conclude for the whole world. The other temptation, even worse, is start imputing like crazy to fill all the gaps in your very, very gappy data set. And this all has costs. So you need to ask yourself whether the extra precision is worth the cost. I would argue that for some purposes, for example, if you want to have a very general, broad brush global picture of something, or if you need to run global models and you need to do it quickly because you want to feed into policy, for example, or if you want to have a very general broad brush picture of the whole phenotype, then the way to go is not digging into increasingly fractal, more detailed spectra. The way to go is using these highly distilled phenospaces, like the ones I showed, and including the global spectrum for vascular plants. Why? Because they act as global large-scale functional maps. So, for example, you can think of the global spectrum of plant form and function like a functional mapamundi. And you can place any species on Earth, or you can follow the trajectory of any plant entity with just 2 quantitative coordinates. And this is based on well-established theory and lots and lots of empirical data. And most models can cope with this dimensionality. So that's why I say that the simplicity of these highly stylized phenospaces is actually their strength rather than the weakness. But of course, if you have very precise questions for a very restricted set of organisms, then this is going to be far too sketchy. And you need unavoidably more traits. In a way, the way I see it is like different approaches to painting. This is a highly hyper-realistic version of a horse by Caravaggio. It makes full justice to this particular horse. You can almost see jumping from the surface and wetting you with saliva. On the other hand, this is Picasso's take on a horse. It doesn't make any justice at all to any horse on the planet, but everyone on the planet would recognize that as a horse because it captures features that are common to all horses. You wouldn't try to sell this horse showing a picture, a Picasso picture of it. It wouldn't take you very far. The same way you wouldn't think of trying to get a Caravaggio-style picture of all horses on the planet, or even all horses on Oxfordshire. It would just be unworkable and ridiculous. So the point I'm trying to make is that these are very different approaches for very different purposes. And the important thing is not to conflate them. So, for example, when one comes up with a very stylized, very oligodimensional global phenospace, one of the big temptations people have is just to start unpacking it with, and never-ending new dimensions to make it more complex and more realistic. Personally, I think that's not a very fruitful path forward. I think that for global purposes, these global stylized phenospaces have to be kept as simple as possible, with as few functional traits as we can possibly get away with. Of course, the very detailed Caravaggio-style inquiries are very, very valuable, but they completely, the purposes are completely different. The importance, again, is not trying to mix them up, and very importantly, try not to pit each other against each other. Okay. So far, I tried to show some of the diverging paths that have developed over the history of the plant functional trait research program, or the just functional trait research program, interspecific or intraspecific variability, Picasso or Caravaggio approaches, and there are three venues of research that I didn't have the time to develop, but I think they are very important and thriving. One is traits measured directly or remotely sensed, links between functional traits and fitness via response functions and demography, or links between functional traits and communities and ecosystem functioning and benefits to people via effect functions. All of these are thriving. All of these are equally valid and fruitful paths. Now, I would like to finish with some blind alleys. Paths that, in my very humble view, are not fruitful. They are not going to take us anywhere. And I will mention two in the interest of time, and also because they are the most obvious right now, but of course there are many more. The first one, I don't know whether you notice the increasing presence in the literature of papers looking at individual traits in response to environmental factors. That's what I call the disembodied traits, because the trait is typically looked at in response to climate or disturbance or whatever, but is considered without taking into account the fact that the trait comes assembled together with other traits in a whole organism phenotype. The trait doesn't exist in a vacuum. And what thrives or dies is not the individual traits, it's the whole phenotype. This trend to work with disembodied traits in my area of the world, we call it the Tupac Amaru approach, in reference to Jose Gabriel Condorcanqui, Tupac Amaru, a hero in the resistance against the Spanish invasion. he was sentenced to be dismembered into different body parts by 4 horses pulling from his body. And if you pay attention, you will see that work with isolated traits is very, very common. I've been author of many of such papers, and they are not necessarily wrong, okay? So, for example, this comes from the work of Ian Wright and colleagues. And what this shows is the predicted maximum leaf area of an individual lamina, depending on a combination of climatic factors. And this is, in my view, a beautiful piece of science. And I feel very proud and very grateful to be a co-author of it. But then one day I was walking at noon on a scorching summer day in the Mediterranean. The air felt like an open oven, you know, in terms of heat and dryness. And there was this plant with this huge phosphorescent green, turgent, tender leaf, being apparently super happy there. While, according to our model, that leaf size in that, under those conditions were very unlikely to happen. But this is if you consider the symbolic traits. But if you start thinking that, you know, okay, is the leaf attached to a whole plant that is annual or perennial? Is the plant erect or creeping? Is the plant woody or not woody? Is deciduous or evergreen? Then the whole thing starts to make much more sense. So the point I'm trying to make here is that I'm not against isolated trait papers, but I think that if we are to predict what is going to actually thrive or die under changing conditions, we need to take them into the context of whole phenotypes. I know that this is easier said than done. And when I say try to look at the whole phenotype, I don't mean measuring every aspect of the whole plant. But for example, when looking at leaf size, for example, having some even cartoonish information of what the plant is. Is the plant woody, perennial, deciduous, not deciduous? That what sort of root it has? That kind of very basic information that you may not even need to measure afresh is absolutely essential to determine what's going to work or not in the field. Second blind alley is about fishing expeditions. Is what I call the pesca milagrosa or miraculous catch of fish approach. And the name comes from the episode in the Bible where there were all these fishermen catching nothing, and then came Jesus and made a miracle, and they cast the nets and they pulled them up heavy, full of fish. This, in a way, is quite similar to what's going on now with the advent of open access big data repositories. Lots and lots of traits there waiting to be used. So people try to get as many as they can of these traits from the comfort of their computer and cup of coffee. And basically theory-free, they start trying to do something with them in the hope that out of the sheer amount of information, some cool patterns will emerge. The problem is that as well as not being guided by theory, there's actually not a lot of fish in the ocean. There is a lot of fish bits. There's a lot of isolated heads and tails and fins. Because even the biggest databases don't have a lot of traits for any single species. Even tri, for example, have very, very few traits on average for each species. So you get all these isolated bits of fish, and what you do, you start gut feeling like crazy. You start coming up with imputed zombie traits, and you put together zombie fish. Now, I don't have anything necessarily against imputation. I think it's super useful for some purposes, especially when you don't impute a lot of your data set, and especially when you are looking at general relationships between traits rather than more specific questions. But when you are imputing a vast proportion of your data set, then, and you don't test how good it is, then you start losing accuracy and precision. And with no theory behind, and with lots of zombie traits, no wonder many of the results are disappointing. But the problem is that when they get these disappointing results, people tend to blame the whole functional trait research program rather than their particular take on it. So I would say that perhaps we need to be a little bit more careful with our pesca milagrosa and start trying to measure some real traits out there, either directly ourselves or from the literature, but traits that somebody has measured in a real plant. The TRI database over almost 20 years, you see staggering growth, but the rate at which the number of requests grows is much more staggering than the rate at which new, fresh data sets are contributed. So I think that in order to make robust progress, this distance has to shrink. And this is a responsibility that has to be taken by the whole community. No group on its own can do it. So some thoughts in order to try to avoid or ameliorate these blind alleys. First, good old-fashioned, be theory-driven. Have a clear theory to test and don't use traits that your theory cannot explain. That would go a long way to avoid the pesca milagrosa problem. Second, try to empirically measure a minimum set of traits that capture the whole plant phenotype, different organs, what the plant as a whole is, even if it's in a cartoonish version. And the third, and last not least, is avoid excessive or uncritical use of imputation. By all means, use it if you need to use it, but don't overuse it and test it before you apply it. And never ever mix, when you release your data to the community, never ever mix your real data with your zombie traits. So if you want to give the whole community your raw data, that's wonderful, but give the matrix of empirical data with the empirical data only, with all the holes, and separately give the code you use for the imputation. So if somebody wants to reproduce your result, they can, but don't put zombie traits together in the same matrix for the public with the real data. One of the things that keeps me awake at night is the awful thought of all these zombie traits released there, never to be retrieved, and contaminating all the empirical data we have assembled for centuries. So, in summary, I think that the way forward is actually by going back a bit, specifically going back a bit to natural history, appreciating it in retrospect, and perhaps devoting a little bit more time and effort to dive into natural history more frequently and more deeply. Thank you.
Thank you, Sandra. We've got time for questions. Anybody
Yeah,
want
can I say something? I have this persistent ear inflammation, so sometimes I have problems with hearing. So state your question loudly and clearly, and don't get alarmed if I get really close to you when you are asking it.
Okay, so you want to kick off? Well, okay, so we'll bang and go back.
Hi, really enjoyed the talk. Thanks very much. So I was interested in this. You showed a slide of intraspecific variation mapped onto the interspecific phenospace, and perhaps it was just coincidental that the three species you plotted are actually from regions of relatively low density at the interspecific level. So that made me wonder whether, if we look at the intraspecific diversity, is that related to your position in the overall interspecific phenospace? Do we see more intraspecific diversity as a function of where you sit in that overall phenospace?
That's a fascinating proposition. I have no idea whether that's the case. Actually, I don't think we have enough data yet to do it for enough number of species, but it would be fascinating. So if I understand correctly, you are sort of hypothesizing that the poorer the area, the more intraspecific spread you will have, and in a very, in the hotspots, you would have very little intraspecific. Yeah, it would be wonderful. I mean, just submit a proposal to try and test it, and let me know.
Careful, next thing you know, you're doing plant ecology.
I had a question linked to the across taxon analyses, different where you show that the body size is a big factor, and then I think you showed for birds, Joe and Alex's work, they're removing body size. You see the other dimensions coming out. I can't remember whose work it was, but so once you take out body size, do you see common Darwinian axes emerging across different groups, or are there different stories emerging in birds and in plants, and in terms of the dominant sources of variation in traits?
Okay, I couldn't tell you about the very new groups like bacteria and ants and fungi. I still have to look at it. In the groups I've seen, I think that in many cases, what I call pace of life, whether you do your life, the things you do in life fast, and invest very little in each thing you do, or you are conservative and do it slowly but imposingly. That's a very universal one, I think. But then, depend on the animal, the third dimension starts to get complicated, at least for me, who I'm a plant person. Yeah, but I would argue that the first two are these, definitely.
Great. Okay.
I think
There's one at the back.
I'll
get this later on.
And I go with you. I go with you.
Thank you very much. Thank you very much for your talk. I really, really appreciate your research, and I wish I was able to join your team when my career was progressing, because it's totally amazing. I worked in the plankton taxonomy, but that was my early stages, and I worked with both phytoplankton and zooplankton, so that's animal, and also in the middle, which is protist world, is like unicellular. organisms that can either have phytoplankton, can have chlorophyll or not. So they kind of just something in the middle. And so here's the question I was out of curiosity. Have you ever considered the space between the plant traits and the animal traits where they mimic each other?
Oh, in what way? I'm not sure.
Yes. So sometimes, like in the coral world,
Yeah.
they would be certain traits that an animal organism would pick up from, or perhaps would there be something like in that
Like branching
space?
pattern, or
That's it, like branching or
right, or I'm getting into trouble here because I know there are animal people here. So I get horrified, they're going to kill me. I would say that I see similarities. Whether one group picked it up from the other group, or whether they belong to a common ancestor to plants and animals, that I wouldn't dare say. I once read a fascinating set of papers by the woman called I cannot remember. She did all this work with very primitive, I don't know, unicellular algae or no, not only. No. I know Lynn. She did these things with unicellular photosynthetic guys, right?
yes.
And she suggested that the pace of life, the conservation versus, you are slow, conservative, big, or you are tiny, acquisitive, and fast living. It was already present there in the very, very beginning of the tree of life.
Yes.
And we all had this, all organisms can follow that. And on top of that, some organisms get bigger than others. And some work I couldn't show you today because it's already too long and also it's not finished, suggests that evolutionarily you have this fast, slow, or conservative acquisitive axis, which is very constrained, very tight, because it gives you very little evolutionary leeway. But body size is like, more freedom, more leeway. So lineages go up and down and come back in terms of size. I don't know whether that goes to your question, probably not, but it's all I can say.
Thank you very
Yes.
much. Thank you.
Thank you, Sandra, so much for your talk. I have a question because I'm quite interested in remote sensing of plant traits, which I think maybe a few people here are. But I obviously see so many ways in which remote sensing could fall into the two blind alleyways that you talked about. So obviously you can remote sense leaves, but whole plant phenology is quite difficult, especially reproductive strategy, which you'd have no idea about. And also, of course, you can map loads of plant traits using remote sensing, but they might be zombie traits as well. So it's quite an open-ended question. So I'm wondering if you could speak a little bit to the opportunities and challenges there.
Okay, I think that there are a few people much more qualified to hear than me to talk about remote sensing of plant traits. But I would say that first, I wouldn't call a remotely sensed trait a zombie trait because there's something real there you are measuring. I call it a zombie trait when you basically do this phylogenetically corrected mean of the whole universe and come up with a number, which you don't have a clue whether it corresponds to an existing plant. So I don't see remotely sensed information as zombie. What you can capture or cannot capture with remote sensing, I confess my ignorance. I mean, I try to learn all the time, and in the lab, I am pestering all of you to look at, to to explain what you are looking at there. I know what kind of things can be sensed easily. I don't know whether you can come up with some kind of a full plant phenotype, remotely sensed composition of data. I don't know. It's a question you probably can answer much more than me.
Difficult.
I hope so, because it's the future. So,
Not reproductive strategy,
Right,
probably.
okay.
But other things, maybe.
then this Rob, yeah.
Is it okay if I ask? Lovely to see you again, Sandra. How are you doing? Thank you so much for the talk. Let me also take a step back to thank you, because your work has inspired many people in the room, including myself, and many people in my lab after that. So I suppose two questions. There's been a lot of work being done at the interface between form and function. And to me, as a demographer, what I would like to see a bit more is linkage to performance.
Right.
And performance is environmentally specific.
Yeah.
And so I often tell my students means mean nothing, right? When we're taking this big comparative analysis at the trait level or at the demographic level, and then we're taking means, we're kind of like throwing the bath water away, right? But I also recognize that we're not going to be able to measure everything under all different conditions, right? So any advice on how to deal with that neglected aspect of these approaches, which is the variance? You've shown very clearly that there can be a lot of variance when measured, when looked for, right? So that's the first part of the question that I have for you. Maybe I'll ask you the second one afterwards. I'll give you room to answer
Okay.
first.
Demography and intraspecific, do you see them as very linked or kind of two different questions? One is, so performance, it means you mean performance as the transition between the individual functioning and the fitness of the.
Yeah. So for instance, you had there your three-dimensional
Yeah.
axis where you had the vital rates of survival, growth, and reproduction, and some traits associated to each of them, right? But
Yeah.
I think, for instance, one that you showed was the couple seed mass, I think it was, and reproduction, right?
Mhm.
But there's a lot more to reproduction than seed
Than
mass.
seed mass.
Seed
Okay.
number, recruitment success, et cetera, et cetera.
Yeah, yeah.
So I think that there's a dimension there that has
Yeah.
not been tapped, and I don't want to open a can of worms and say we should be measuring more traits, but I think that we're missing a lot. If we really want to build a castle to go from form to function to performance.
Okay. Form to function of an individual, I would defend that the minimalistic approach, taking very few functional traits, so traits at the level of individual, it takes you a long way. Now, if you want to know the performance of populations, how the population persists and grows or dies, 100% in agreement with you. We should be looking at more demography, probably different communities of people, because typically the expertise needed.
And we don't talk frequently enough, right?
No, exactly. And the expertise needed is very specific and very rarely comes in one person. The problem I always had with demography, that you may have some answers, is that when I try to look for how many of my species we have full demography data, is very low because taking demographic data is just so painful. So I don't know what you would propose. I mean, how far are you from having, many species?
It's orders of magnitude lower, and I think you put the finger on the one, right? It's perhaps more time consuming. I'm not saying that traits are easy to collect, but we can talk about that later.
Right.
We're developing some shortcuts with
Well, that would
remote
be
sensing, actually, among others. Yeah,
that would be wonderful. And you had another question.
Yeah, but I don't want to dominate the conversation, so I'll ask you over drinks, actually.
Maybe I can take it since I'm going to get close. Okay.
Okay.
Yeah.
Hi, Sandra. Thank you very much. This was really inspiring. Perhaps a simple question. So you had this paper, right, with the shapes and with planes, the two planes, and everything like this. You have these two main groups, like just that together, and just playing the traits that are being super important for those two planes. So is there kind of, have you thought about what is actually driving these two planes? what would be kind of a simple mix of environmental conditions, perhaps, that may be causing these two simple groups?
You mean
Uh,
the hotspots?
Yes.
Well, the hotspots, I didn't want to dwell in the global spectrum, but they are herbaceous plants and trees, not shrubs. The shrubs fall in the south. So yes, I mean, there's no geography in that ordination. There's no geographical reference in climate, nothing. So in that in the hotspot there were all trees, mixing trees from everywhere, but very strongly dominated by tropical, because the tropic is where you have the vast majority of tree species and the tallest them, right? And then the herbaceous one, then you have herbaceous little things from all over the world, because there you have mixing, you have dicots, you have grasses, you have micro shrubs from all over the world. But I have some work for, I've seen two new ordinations. One was published last year. It was for Canary Islands. I helped in a project for the Canary Islands. Paola Barajas Barbosa is the first author. And this is, you get it, because in the global spectrum, you don't have environmental filter because it's the whole planet, right? But if you get the Canary Island flora, Tenerife, then you have a strong climatic filter. And it's quite interesting that the global spectrum is pretty much the same. It's like a micro version of the global, but the hotspot is only one, and it's in the shrubs, precisely where this global saddle is. And I just seen data from Eugenia. You met Eugenia a couple of years ago for the Argentinian, the central Argentina flora. And also you have a lot of filtering there. And you don't have the tall tree hotspot at all. You just have one hotspot, which is the herbaceous, but shifted to the really conservative side. So I would say that this is the global catalog of plant design, if you allow me the metaphor, the one I showed. But depending on the local filter, you get the micro hotspots shifting around. Yeah. Yeah. Oh, okay. I think he was first, and then I come back.
Thanks very much. So last thing on a Friday is a nerdy question. So you're cheers. So you're analyzing your data through principal components analysis.
Yeah.
That's how you're generating
Yeah.
your clusters. Have you looked at other methods such as independent components analysis?
No.
Okay.
No, we tried Varimax, and that was when we did this first analysis. We also tried to be minimalistic there. And we say, okay, what is the most well-known and simplest method? PCA. Everybody knows PCA. It was perfect for the kind of data we had. Some of the members of the group wanted to try Varimax, which would have straightened, it's kind of diagonal, our plane. Varimax would have straightened it. But then we had two multivariate nerds, people who do, only multivariate statistics. And they had all these terrible objections about Varimax. And they said, if you use Varimax, I walk out, for really arcane reasons for me. They just didn't like Varimax. And they are, the reasons they had, it was strongly mathematical. I couldn't possibly, I just had to believe them. So yeah, I don't know what it would be. I would say it shouldn't change a lot.
Okay. Would be interesting to see what.
It would be interesting to see. Well, I mean, yeah, it can be done.
Okay.
Yeah.
Thank you. Cheers.
Yeah.
Well, thank you for the talk. So when you do this projection, that projection that would be influenced by big families with big variation, the optimal projection. Isn't there a risk that small families or endangered species would be overlooked because they will not contribute much to variation, therefore they will not really change this projection and they will just kind of be hidden?
I'm not sure I understand you. I will try and answer, and if I'm not answering what you are asking, you ask me again.
Okay.
Yeah. At the global level, the amount of phenospace occupied, is not very well correlated with the size of the family. For example, the typical example, you have tiny families, which are all, the cycads. The cycads are all in a tiny place. There are very few, and they are all the same functionally. And then you have a huge family like the legumes. The legumes occupy almost the whole of the phenospace. But then you have the orchids that are, I think, is more species than the legumes, and they are all in a tiny little bit of the space. So it is at the global scale, it seems more or less independent from size. So I don't see the risk. I think that some families are more diversified than others, and yeah.
Okay, I'm not talking size and like physical size, but more
I'm talking about the number of species.
Yes.
Yeah.
So if there's big variation within the family,
yeah,
that would cause them to spread out in your trade space, which will then dominate the prediction.
Okay, let me see if I understand. You, for example, suggest that because the legumes are very many and they are very functionally different, they overwhelm the ordination. Maybe, but I mean, there is a reason why there are so many, right? So we felt that sampling to say, okay, we have the same number of species from every family on Earth. It's an approach. You will have a more taxonomic control, but I'm not sure would be closer to what's going on in the real world. But I'm an ecologist. I'm not a phylogeneticist. So probably our approaches would disagree depending on our area of work. Yeah.
Thank you.
Hi, Sandra. Thanks a lot for your talk. And my question is about what do you think it's the role of the CSR theory in the future of functional ecology?
Oh, okay.
Because, well, I mean, I'm a PhD student,
Right.
and I'm supervised by the Professor Bruno Rosado and Erika Beringer.
Yeah.
And I've been working with functional ecology and plant hydraulic strategies in tropical forests. And one of the approaches I use is the take as a first step, testing the predictions of the CSR theory.
Right.
But what we have that sometimes the CSR theory are not enough to explain some variability, especially at smaller scales, spatial and temporal scales. So in these smaller scales, hydraulic traits are better. So I would like to know. Hydraulic strategies. Hydraulic,
yeah.
sorry.
Oh, yeah.
And so I would like to know what do you think about the role of CSR theory in the future of functional ecology? I mean, with this new, more integrated traits, which sometimes hydraulic ones are like plant sample flow, and there is a kind of hierarchy of traits we have. So I would like to know, what do you think about that?
Okay, that's quite a question. I think that the CSR theory is a strategy theory. You know, you have a prediction of what the whole plant does, right? This was a much more, I talk about syndromes. You probably noticed that I didn't talk about strategy.
Yes.
because strategies is more, more teleological. You say, well, this is the game this plant is playing at, and therefore it should look like this, right? This is, or you could do KR, another example of strategy. This is more agnostic. You say, okay, these are the syndromes. We are not saying anything in, committed on what they are playing at. These are the syndromes. So these syndromes are very compatible with the CSR theory. I mean, you could, if you wanted, you could draw the Grimes triangle somewhere there, or you could draw the RK there. Now, that in general, so it's not incompatible at all. I think that many people like CSR. I like it long time. I still like it. So it's it's not like it's an outdated theory, right? Having said that, I think it doesn't do super well with really, really big and slow growing plants. Like, I think that, I mean, I love the CSR theory, don't get me wrong. I agree with that. But I never, in the global spectrum, it works perfectly with the first half. If you take the herbaceous world of the global spectrum, then you can draw easily the Grimes triangle on top of it, and it would work beautifully. But it doesn't do very well in the other part, really big, slow growing things of the tropic, or really super conservative, but also super drought resistant guys. You are right that the theory was never very good for hydraulics because it was never very good for drought. You have to remember that it was born here in England, right? And no matter how general you want to be, it permeates your idea of the world. And here, I mean, to be honest, water is very rarely a problem. You have nutrients as the major source of stress and disturbance. So that would be my take. I'm not sure how well would work for tropical forests of Cerrado.
Okay.
And the problem I see, but this is not a problem of the theory, it's a problem of the method now.
Right.
Simon Pierce's method. I am a co-author, so I like that paper.
Yeah, so as far as I know,
Yep.
that your paper was an inspiration for the quantification.
But the problem I saw, and they, Simon and Francesco and all of them fully recognize it, as the traits you enter into the software to come up with a strategy, there's no body size. It's all leaves. And, it gets very good for some plants, but you get utter rubbish for others because the woodiness and the final body size is not there. I mean, it can be fixed, I guess, but I don't know how to fix it, but I'm sure the main authors of the paper may find a way.
Okay,
Yeah.
thank you so much.
There was, and there was somebody else before? No, okay.
I have a very quick one. So just now you're explaining on the global PCA, and you have like herbaceous here and woody traits here.
Yeah.
And to what extent the middle gap could be possibly filled by liana and epiphyte?
By
liana
Oh.
epiphyte.
Ha,
Oh, no, you don't need
brilliant. Okay, I may have to project the slide again. It won't take long. It won't take long. Any of the, yeah. Oh, I mean, that, yeah. I mean, ignore the intra-specific, please. I would say here are mostly shrubs. right? And shrubs, and this is not a limitation of the global database. We did all sorts of tests to try to see whether it was an artifact of the data set. It's not. But then with the Barajas Barbosa paper for the Canary Islands, as I said, the whole flora of the Canary Islands is basically here. Not the whole flora, the hotspot is here, the shrubs. The lianas, I mean, we do have some lianas in the global data set, are so all, on average, they are super acquisitive. You know, they don't have to spend any resources in support. So they are here. Depending on who they are, some of them, like the woody lianas are here. And then the ipomeas, you know, ipomea, really flimsy. You probably don't have it here. Morning glories. Morning glories are here. So all this area, which is a cold spot globally, is occupied by the liana. And the epiphytes depend on what epiphyte you are. The data I'm just seeing in central Argentina, we have a lot of Tillandsia, so bromeliads, really strongly conservative bromeliads, and they are here pushing the very border of the spectrum. So they are not in the middle; they're just in the cold valleys. Yeah. Okay.
Last question.
Yeah.
Thanks, Sandra. More of a conceptual question, I guess. So you showed like these are the important traits as of now. To what extent do you think it could be biased that maybe there are other traits which are not really measured that much? And is there a possibility that could be biased? Another thing is like with climate change, do you think in future, maybe some other traits might become more important than these? Like with it's becoming more drier. So maybe, do you think there's a possibility that there will be more traits which become much more important than this?
Okay, at the global level, at the world level, remember this is just the whole catalog, right? I don't think, I mean, as again, you can add dimensions with other traits. Like we try with the fine roots, and they added a little bit, you know, a little bit of a thickness to the chapati, right? A little bit. And I'm sure if you put some hydraulics, you will add a little bit more, and you will kind of fractally add dimensions. I don't think, I may be wrong, but it would be hard to imagine for me that size and pace of life wouldn't be the major dimensions. I mean, the reason we chose those 6 traits is because, okay, they are very available, but there's a reason why they are available. They are really capturing very essential things. So I don't think they will become, you know, bottom of the list and, you know, flower color being the most important one. I cannot think of. And global change obviously can change the topography of the landscape. Like, I mean, you can argue that over evolutionary history, there used to be a hotspot here, all the gymnosperms, right? And for some reasons, because of climate and also because of disturbance and everything that happens in, you know, in the quaternary, this is not a hotspot anymore. And then you have hotspots that weren't hotspots in other geological eras. So you just fast forward and you may have a shift in hotspots. That's what I would love to do, and I'm trying to do, to see how are we altering the topography. I don't think we will do anything that will alter the boundaries. I mean, we could do it by genetic engineering, but other than that, I don't think so.
I wonder whether the hotspots and the diversity, and the number of species in there is is changed that much, and whether the extinction of the Jurassic gymnosperms has really changed the total diversity of gymnosperms and forms. Probably not. I suspect it's probably just meant that they're not abundant in the same way as they were there. But
So your point is they were just very
I
abundant, but
suspect-
maybe they weren't many species.
Yes, yeah, just like the polar forest now.
Yeah,
they're abundant, but they're not many species contributing to this
Well,
diagram.
I was told, but I don't have my own opinion. I just told, because it's not my area, by paleontologists that there were many more gymnosperm fossils than the number of gymnosperms we have. Once you calibrate by, corrections for fossils that get lost, I don't know. But again, I don't know whether it's true. They say, but I don't have my own qualified opinion. Okay.